Thanks so much. It's a pleasure, and an honor to be here. We're gonna, try to talk about the impact of nanobiotechnology, and as mentioned by the chair here with me, that clearly, nanoscience, bring all facets of science together. You're not gonna find that, as good as in nanoscience. Physics, chemistry, polymer chemistry, pharmaceutics formulation, drug delivery, pharmacology, biotechnology, cell biology, even business development, regulatory affair, marketing, especially if you want to take the whole concept all the way from the bench to the bedside. That's usually what I do. So I like to do the science that matter at the end of the day. So, and we're going to try to touch briefly on precision medicine. Actually, I'm building, two certificates at the College of Pharmacy in Albany, one on nanomedicine, one on precision medicine, and combine the two together, according to New York State, that will lead to a master degree in professional studies. And this will be online and in many different facets, hand-on experience, workshops. So, this is for the young people. If they are interested, we will be happy to help them out. I just want to make clear, a statement here about the impact. If we look at the Industrial Revolution, it took about 200 years to realize its full potential. If we look at the molecular nanomanufacturing, the nanotechnology, it can take, it can approach the same potential in about 1/10 of the time, in 2 decades. And I have seen in Albany, by the way, you have to visit and look at Albany. Albany is the capital not only of New York State, in terms of nanotechnology, is the capital of the world. And I'm going to tell you why, because this is a big statement to make that statement. In Albany, the state of New York funded, they put about $1 billion. There is an enterprise now in terms of using semiconductor and nanobiotechnology consortium. It is about $12 billion enterprise. If you come to that site, it's almost occupy Washington Avenue there is about 5,000 people employed at that site. Actually, Obama, if you want to brag about nanomanufacturing and nanotechnology, they have even sites within the whole New York State. He comes to the Albany they call it Albany Nanotech. There is about 240 spin-off companies out of that. The facility and the resources is magnificent beyond my description to it. You're going to find the equipment that's being shared by Intel and Siemens and GE. The cost of one piece of equipment, it could be $2 billion cost. It's just unbelievable. One company cannot afford it, so they share. But they have time, and they have protected time, and they share the confidentiality. Everything is. So somehow just go and look at the website and so Albany Nanotech. It is the capital of the world of nanotechnology in Albany. So I will be happy if you happen to come and visit Albany. I will give you a tour to that facility. But this is just to show you, and actually this happened in 10 years. In 10 years only, from 2002, $1 billion, it led to $12 billion impact. And you can see the value on the economy. So clearly there is no doubt about it. So what are the 5 top applications of nanotechnology? Since I'm the first speaker here, of course, nanomedicine, actually has a major, major application for nanotechnology. Drug delivery systems, diagnosis, screening, monitoring, lab on a chip, tissue engineering, healthcare delivery. Also energy, storage and production and conversion. Nanotechnology have a major impact because of the material, exactly like what the professor said. Agriculture, productivity enhancement, and food processing and storage actually it's currently being employed. Water treatment, air pollution, and the remediation. Supercomputing. The wafer now in Albany, it is 450, it's just unbelievable, the amount of information that you can build a very small, like your cell phone, and the capacity. All of this has actually been done in Albany. In a way, the chips, nano chips. Plus, there are other applications in construction, textile, and equipments. The equipments, in a way, actually is something which I used to complain about when I was a graduate student. The bench space available becoming smaller and smaller because the equipment occupy a huge bench space. Now the equipment are going to shrink and going to be smaller, so we are going to have more bench space to work with. All right. Nanomedicine. What's nanomedicine? Because it's a medical application of nanotechnology. And I'm going to convince you that nanotechnology alone is not good enough by itself. You have to bring a lot of technology to make it valuable, make it applicable. Nanomedicine range from application of nanomaterial to nanoelectronic, biosensors, molecular nanotechnology in pharmaceutical, biopharmaceutics, in even nutraceuticals, and cosmetic, nanocosmetic, nanonutraceuticals, diagnostics, and other future healthcare application, even healthcare delivery can benefit from nanotechnology. It is about nanotechnology is, as I said, in standalone is not enough. You have to integrate it. It's about integration, technology integrations. With biotechnology, you need bioinformatics. If you use biotechnology, you are going to generate a lot of data. So you need to sort out this data and find out the key parameter that you need to take home and run with the applied to nanotechnology. You need the traditional pharmacotherapy, pharmacokinetic, pharmacodynamics, and pharmacogenomics for personalization. Very important component here. Stem cell actually will benefit greatly from nanotechnology. You can build nanoscaffolds to implant the stem cell, and then you can put signaling molecule in the scaffold to make the stem cell differentiate into whatever, to myocytes, to endothelial cells, to bone cells. So this is, we can have an implant, bone batch, to repair the bone back again. Tissue engineering will benefit greatly from nanoscaffold and nanotechnology. And I showed you that slide, I think I already talked about it. So why nano? What is the secret of nano? Nano is, according to the Greek, from the Greek definition, it's dwarf, and which means 1 over billions, which is 10 to the minus 9 in terms of meters, 10 to the minus 9 meter, which means equivalent to 1 nanometers. So it's a nanometer scale size. But very important, I want to just keep that statement in your mind. When you make the size of the particle in a small size, the surface area increase. That has the physical properties of the material, tremendously improved. So now, and I will show you application and FDA approved the product and how it made a difference from the traditional material or even broad chemical into a nano-sized or nanocrystals, making the material nanocrystals. Now you can improve the bioavailability, avoid the food-drug interactions. You can have completely different profile, improve the profile for the product by making it a nano-sized. Because of the physical properties in the surface area, it changed dramatically. And this is relevant, water is 1 nanometer, glucose about 10 nanometers, antibodies around 100 nanometers. And you can go virus is about 1,000 nanometer, bacteria about 10,000. Cancer cell is about, almost, 1 million. And a tennis ball is about 100 million. So usually the nano size, the definition, I don't care about the definition between you and I, as long as you come up with a lot of people argue about what is the definition of nano. Some people say it has to be 100 to 200. If you can bring it less than 1,000, I'm fine with it as long as you can make a major improvement in the application of the product. This is my application, my really definition. What matters is can you transform a product to make it a better product by nano-sizing it. There is a lot of argument, and actually, but recently they settled the debate by saying anything less than 1,000 nanometer. It's a waste of time for people to argue and debate. The key is in the matter. I'm a very practical scientist. Can you transform the product and make it a better product, applied product, even if you make it 1,000 nanometers? They have to compare the product before and after. Did you make a major impact? Did you come up with a better profile? So why nano is important? Traditionally, if you give a pharmaceutical or a drug to a patient, it goes everywhere. But the nano, usually, if you do targeting into the liver, for example, or into the pancreas, or into the tumor, you can target the liver into the site of action. And here is a sketch of a nano shell. The nano shell, inside here, you can encapsulate, say, chemotherapy, multiple antiviral drugs in combination. You can have on the surface here, coating on the surface modification, and you can conjugate a targeting moiety that is specific for launching that nano product, the nanoparticle, into the site of action. You can have an imaging probe. You can put actually DOTA, and you can put gadolinium, and with the targeting moiety, you will be able to take that nano into the site of action, and you can image it. So you can image and treat at the same time. That's what they call it, thronistic. And clearly, if you can do this, you should be able to improve the advantage of nano, improve solubility, stability, because you hide it inside the shell, specificity, multimodality. You can have multiple drugs inside, and that will improve the efficacy. And then at the same time, if you can deliver it into the site of action without getting everywhere, you should be able to improve the safety profile. What is the application of nano? Drug delivery, gene therapy, and imaging. The assembly here is you can form it using, again, a polymer, for example. In that case, a PLGA, copolymer polylactic glycolic acid. You can see the acid in the outside, the polar heads, and the tail is the lipophilic one, and that's a regular micelle formation. And you can incorporate hydrophobic drugs inside, or molecule inside the core. And you can do reverse micelle by reversing the process for incorporation of a hydrophilic compound inside the polar region here. And the outside, you can now use stabilizers to stabilize the nano. You can put a cryoprotectant to prevent the aggregation of the nano. These are manufacturing process that you will be able to do. It's actually, the manufacturing is very straightforward. So there is a lot of actually great opportunity in a way to do nano manufacturing. But the key is in the assembly, and the art is basically in using the knowledge base to try to target, deliver into a specific application. And you should be able to make a major impact or major unique applications relative to the existing applications. Opportunity for nanotechnology in drug delivery, clearly, it's a solution to many insoluble drugs. Big pharma usually, or many other, whatever you have an insoluble drug, that makes it difficult, to introduce. The bioavailability will be very low, very poor, especially very hydrophobic compounds. That you can fix it with the nano. Stability, something like a peptide, you can now hide it inside the shell. And now you can protect it from the degradation. And maybe you can actually design the material where it breaks down only with a specific enzymes in the location where you need it to deliver. So there is a lot of art and science in this. Rate of dissolution, you can control rate of oral bioavailability will be improved. Food drug interaction will be definitely improve a lot. Targetability, you can lower the dose administered. You can actually minimize side effect. You can have more convenient dosage form. You can improve the efficacy, like what I mentioned before. Again, I just want to emphasize this because I think this is black. There is a black here. Sorry about that. That is not showing. But the bottom line here about nano, increased surface area and more physical properties. You can actually multiply and enhance the surface area of the material. I'm going to show you examples here because sometimes some of the people usually say, is this nanomedicine is a science fiction or is it a reality? Actually, it is happening. And even the word nanotechnology, it's basically existed way before, but nanotechnology, the word nanotechnology attracted an attention. But if you look at colloidal, nanocolloidal, the colloids, these are nano. This existed way back. But as I said, the bottom line for me is in the application. Am I going to make an application out of this technology or not? If I can't make application out of it, then it's interesting science. It's just interesting. But the bottom line is, how can you make significant, unique innovation out of this? So, here is a first nanoparticle suspension product by Weiss, which is an rapamycin immune suppressant. Actually, the way they launched it to begin with, it contained all of these mix here to make it in a nano size, and that enabled them to produce this rapamune tablet. But there was issue with that first formulation. So reformulation of the Weiss product improved the bioavailability by 27%, and they improved the compliance. And this is a tablet of the rapamycin tablet here. So and they provided, of course, big pharma usually when the patent expire, a year before, basically they try to scratch their head because the revenue will just dip down. A lot of people will get laid off right there. This is a byproduct of patent expired. So the question, how you can manage the life cycle of the product and how can you extend the lifetime of the product? So, nanotechnology actually is a life-saving for the life cycle and for extending the patent. And you look at another product here is in a capsule. This is a nanoparticle suspension substance P antagonist. This is the nano suspension here, improve the bioavailability, reduce the fast food variability in bioavailability. Very big problem if you are a pharmacokineticist, do pharmacokinetic studies. The food, interaction, and you give the people, they have to be fasting, and then they eat 2 hours after. If you can avoid this, the compliance would be increased tremendously. This is, so you say about how technology can, improve compliance and minimize the inconvenience to the patient. Here is a marketed nanoparticle suspension product, and they filed patent, and the basis of filing here, and the patent got issued, and the product with lifetime was extended, efficacy and safety or pharmacokinetic improvement. Here, another list of example of nano-sized active pharmaceutical ingredient nanocrystals that actually was approved by the FDA and made it to the marketplace by many sponsors here, as you can see. So I'm not talking about science fiction. I just want to convince you by that, because some people, sometime outside the domain of the nanomedicine, they think this is, when it's going to happen? Is it going to happen 10 years from now? It's happening. It happened, actually, and it will continue to happen and progress. And, like the fast train. The fast train is, so you have to jump on the train. Otherwise, the train will leave you, and you can stay on the station scratching your head. So this is what I tell the people who are skeptical about nanotechnology. There are a lot of products, and there are many products moving very fast into the clinical trials. Liposomes formulation approved by the FDA. Here is a list of them. Okay, the question comes here, sitting here together today. So these are products that are already launched and making a lot of money. So, the question that we need to ask ourselves, drug discovery and drug development is a very long-term cycle. It takes, a shorter time for discovery, but the development cycle can take about 10 to 12 years. If you think about the time to file your patent, you get 20 years. You have 12 years is taken away, so you have only 8 years, if any. As a matter of fact, you might have only 5 years left. And then you need to make money to recoup the cost. So the question I asked myself, and a lot of things can go wrong here. This is a high risk. I lived in that side of the world for 17 years. And, so there's a lot of art and science here. How can you avoid, the failure and and enable the technology to accelerate and shorten the time to drug discovery and drug development. So I asked the question, could this enabling technology, nanobiotechnology, shorten the time to drug discovery and drug development? I can tell you the answer is absolutely yes. How can we do this? I'm going to show you examples. We can take existing drug, the safe bet here, take already FDA-approved product. They are generic. And what you can do, in a way, as a plastic surgeon, put a facelift on this product. In a way, make them look nicer. A 70 years old, in a way, person, make him look like 50 years old. We can do that with the nanobiotechnology. And we can actually improve the PK and PD of an existing product that's generic, the patent expired. I'm not saying this as an abstract. I have a lot of patent, IP issued in that domain. Anyway, so I was able to convert injectable heparin into a solid dosage form heparin. Heparin is a polyanion, negatively charged. It cannot cross the GI. So can you do that with low molecular weight heparin? Yes, you can. Anyway, so what is the, and I'm going to show you this snapshot of examples. I can show you a lot of examples because of that. My time is 45 minutes. Anyway, BD, you can improve the pharmacodynamics. Insulin is also, you can actually package it and protect it. And even on a first pass, the best route for administration of insulin is oral, because the hepatobiliary system is the first pass. This is where you need it to get. Injection of insulin is not the right route of administration. The right route of administration to get it to go on a first pass into the pancreas, the hepatobiliary systems. You can do combination of multi-Rx, chemotherapy. You can do antiviral combination of for HIV, for hepatitis C. I have patent on the HCVs. You can target them to the virus, as a matter of fact. You can target them to the hepatocyte, to the liver. So these can be reality, targeted delivery. You talk about, how about new, if I'm working with a new novel drugs, for example, I'm working on a target called PCSK9 inhibitor. This enzyme is in the liver. If you are genetically have high expression of the enzyme, your LDL cholesterol will go to the roof, familial hypercholesterolemia. So there are 3 antibody FDA approved, 2 antibodies FDA approved, 1 is under review now. Amgen and Regeneron and Sanofi. And then Pfizer is having 1 under review. So 8 years ago, we worked on developing a small molecule to make a tablet instead of an injectable, or monoclonal antibodies. So where is the target for this? Where is the enzyme? The enzyme is in the liver, in the hepatocytes. So we can actually take that small molecule, package it, direct it to the hepatocytes. So you can target the enzyme where it is the origin of the enzyme. And we can suppress the enzyme activity or the interaction between the enzyme and the LDLR receptors. If you are in cardiovascular, you will understand what I mean. This enzyme degrades the receptors, and when you degrade the receptors, the cholesterol is usually shuffled from the circulation via the receptors, the LDLR receptors, to be catabolized in the liver. If PCSK9 activity is up, the receptor is degraded by the enzyme. You have no receptor, you accumulate cholesterol, like familial hypercholesterolemia, or many normal people like you and I have relatively high expression. We have high cholesterol. So this is can be used in combination in a solid dosage form with statins. You can improve the PK and the BD, and even for biosimilars. So you can actually bring new biosimilars. You do target delivery. You can actually, if you want to get genomic action versus or you get non-genomic action, you can do the imaging. Imaging is a huge field. I spend a lot of time in big pharma in imaging. This is, the multifunctional, polyfunctional ligands on the decorating the surface of the nano surface from the material. This gives you a high affinity, positive cooperativity of binding. See the affinity, specificity, and the detection, the early detection can be highly achieved. And then you can put a core of a DOTA with gadolinium. You can put copper 64 for bit imaging. So you can use all of the existing modalities. This is what I mentioned to you. Can we convert heparin injectable into an oral heparin? I'm not going to show you a lot of the composition here because this is better. This patent is pending. A lot of patent in that regard. Heparin is a polyanion, a negatively charged compound. You can actually even depolymerize it to make, this is the pentasaccharide. I'm a thrombosis guy, so this stuff is actually close to my heart. And you can actually slice it into different fragments to make low molecular weight heparin. And these are very good anticoagulant. They have to be given Heparin is given by IV injection in the hospital, low molecular weight heparin subcutaneous, so people can inject themselves at home. But they are negatively charged. Negatively charged cannot cause a placenta. So what can we do? This is actually a trial that we conducted and published. Heparin even tried to do a permeation enhancer. So you can see the value of nanotechnology. Oral heparin here by using sodium caproate. And this was actually a small startup company. The bioavailability at the end of the day was about 2%, not good. With an anticoagulant, this is very low. You need the bioavailability to be not less than 20%. So what we did here, we did a lot of formulation in a way of low molecular weight nanoformulation. I'm not going to disclose the nanoformulation for you, but the concept is very simple. If you look at the polyanion, we can hide it inside that shell. And then we can coat the shell, functionalize the shell, and make sure that the shell will never open in the GI. Or in a way, it will give us sustained release as well. So this is subcutaneous dose of tenzaprin, low molecular weight heparin. And here is two different nanoformulation, the red line and the blue line here of tenzaprin, a low molecular weight heparin, FDA approved. And now we can get a significant improvement in this by the PK profile of a subcutaneous product. How about the oral? Here is the oral. I don't know if you see it from the back or not. This is a black line here. This is a nano-tenzaprin formulation. If you give tenzaprin oral, the red line, you have zero bioavailability, day and night. So this is, in a way, a novel nano formulation, and we can control the release kinetics. And now you can lower the dose, you can sustain the anticoagulant activity, you don't have the peak and trough, all of the stuff. Okay, other applications. Silver in our nanoparticle, metallic nanoparticle. You are going to hear a lot of metallic nanoparticle in this conference. We have done silver nanoparticle, gold nanoparticles, but this is a silver conjugated again to my favorite heparin-derived heparin product. And when we conjugate this, we end up with a nanoparticle that have the size of 7 nanometers. So, and this is a patent that was issued. We have a lot of publication in this. And now, for me as a practical scientist, so what is the big deal about silver heparin nanoparticle? What can we do with it? So we thought, being in the thrombosis area, people usually, if you look at the publication, they talk about catheter. When you indule a catheter especially pediatric cancer patient and elder population, hemodialysis and so on, the catheter usually will get clotting and get infection. So what silver is good for is a very non-specific antimicrobial. It's a polycation. And the bacteria, the bugs are polyanion, have a negative charge. So it will create a magnet. You will actually attract the bacteria and the bugs circulating in the catheter, and we can coat the catheter, embed this in a cellulose membrane, which we did. We published that recently. Embedded, with doesn't leach, and now any bacteria, any bugs that will go through it will be adhered to the silver and will be lysed. The heparin will prevent the clotting. There will be no clot. So the catheter will be patent for 28 days. You don't have to, because what happens when these subjects get clotting or infection, you give antibiotic to the subject. You give IV heparin to the patient. So you create more complication, more than what you want. See, the problem is a local problem. So the solution for this local problem is a local solution. And using something like this literally solve the problem. So now we are moving this forward into a commercialization. But I just want to tell you, simple concept like this can have a lot of impact. And there are about 1,000 papers published in the literature every year on catheter-associated infection, catheter-associated clotting. With this kind of platform, there is no need for this publication. We are actually better off without this publication if we can get rid of the problem at its route. Just simple example. I say when you are in nanotechnology, you can't say I'm only in nanomedicines. So we stumbled on a concept here I will share with you, but I'll give you the background in terms of the nanotechnology application in textile and rugs and carpet manufacturing. If you look at many of the fabrics, There is a lot of nanofibers already made clothes, water repellent, wrinkle free, and surface protection from electrostatic starch. This is already exists. So we were working on a musk oil. Are you familiar with the musk? Okay, the musk is very nice fragrance, and it does have a very powerful bioactive antimicrobials at the same time. So we're able to make a polymer out of that musk oil, a polymer like nanomaterials. And now we made nanofiber out of it. We patent this. And then you have the antimicrobial property, you have the fragrance. Actually, this application in textile, in rugs, look at we are sitting here in the hotel, we have rugs here. These rugs collect with the humidity, especially in the Texas area, a lot of humidity, fungus, bacteria can grow. If you have this, along with a nice fragrance, you can control the level of fragrance, you will have none of this problem will happen here. So you can actually have a very sterile environment that you live in. But this simple concept that we stumbled on by accident, as a matter of fact. All right, how about vaccines? We're looking at, I usually like to look at the problem, I'm a problem solver. Looking at this hepatitis B vaccine. Hepatitis B vaccine, I have a lot of people in my R&D institute from India, as a matter of fact, from all over the world. China, from everywhere. And so, I ask about what is the problem. So the rural area, they have to have a 3-post booster from this vaccine. The alum, the antigen here for hepatitis B, and then in an alum formulation as a new adjuvant. The key, they have to get 1 boost. A month later, they come back, or 3 months later, they come back for the second and the third. I said, so the whole idea is to keep the antigen circulating for a long time. And then you don't have the patient to come. Actually, most of these people, because they are not educated, they are poor, they don't come back. So they don't develop the titer or the antibody that will protect them against the infection. So we developed actually a novel, a chitosan, with the antigen, and we devised this. It is a little bit complex nanoformulation. But chitosan, was enabled us. And chitosan, by the way, is a good material. It comes from the shellfish, or actually I favor the plant source from mushroom. And there is a GMP manufacturing facility in Belgium. That's why they come to Kennett Square in Pennsylvania, and they contract all of the mushroom to be shipped to them in Belgium. But it is a very good source, and we were able to look at the titer. This is the antigen alone. This is the alum antigen. You can see the titer is building up. And then if you now formulate it in chitosan, you can have a very sustained generation of titer. And this will be 1 boost instead of having 3 boosts. Using technology to solve problem and avoid actually a lot of unnecessary issues in these patients. And this is reported so you can look at the paper in detail. There are multiple papers on this, but this is one of the papers. Now I'm going to move to cancer, because nanotechnology has a major impact in cancer. I just like to remind all of us, in many diseases, I worked in cardiovascular, and my favorite meeting as a cardiovascular person, American Heart, American College of Cardiology. I'm a fellow of the American College of Cardiology. And we were able to make good advances. The risk in terms of morbidity and mortality reduced by more than 50%. Good medications. Hypertension, antihypertensive, antiplatelet drugs, anticoagulants. Major advances here. And actually, side effect is not tolerated. If in the American Heart, if a patient have coughing, this will be actually a red flag. We don't want side effects. Even minimal side effect is not tolerated. You go to cerebrovascular, there is good advances. There is pneumonia infection, there is good advances. Cancer, this is from almost 1950 till almost 2010 or 2008. No differences And add to it, there is a number of new cancer cases will be increased by 30 to 50% as we approach 2020. And a lot of money is being dumped over these years by NIH and many agencies to do entertaining science. Literally, I call it entertaining science, good publication. The ultimate, I publish a lot. Don't take me wrong. I have the highest publication record ever. But at the same time, I patent first, and I publish. But also my eye is on the finish line. So, the ultimate goal for academic, that I'm trying to teach academic scientists, I'm the head of research over there. I said the ultimate goal should not be only just getting publication. This is a lousy goal. The ultimate goal should be how can we take our science to really make an impact on human life. That should be the ultimate goal. The money is is almost like a catalyst. It should not be it is a mean to do what we need to do, but should not be the goal, to be obsessed, but I need money. That's why I'm talking about the economy of experiments. How can you do your experiment, which you multiply your money to go a longer way for you? So and at the end of the day, your ultimate goal is to make an impact. But this is what it is. Lucky there is more advances coming in, and I believe that nanobiotechnology will really make a very important difference, especially with this kind of statistics. We should be able to make a bigger impact in cancer. And I'm not an oncologist. I got into it because I got into angiogenesis. If you got into angiogenesis, you found yourself landing in cancer. And I'm a vascular person. But here is the nano platform that we usually try to work around. I like to, this is a sketch. This is a nanoshell, a material. You can use a lot of material. You can use biodegradable polymer. You can use metallic nanoparticles. You can use, there is a lot to choose from. But you have to be very conscious about the safety of the material. And, because at the end of the day, if your drug is the most effective but unsafe, that will not cut it. So, so you can functionalize the surface of the material. You can have a, targeting moiety. This targeting moiety could be a small molecule for, say, integrins, for specific CD that's being expressed on the cancer cell or the cancer stem cells. And then you can have an imaging probe here with data, but you have targeting. Targeting will help you to get the imaging into the gene. You can encapsulate inside a chemotherapeutic agent, multiple chemotherapeutic agents. So this is multifunctionalized nano. This is what they call it thronistics, but it can be even far better than even the nomenclature of thronistics. So this is an example of multifunctional approach for treatment. So you can have a drug that acts. You can have a suppressor of drug resistance. You can have suppressor. You can actually destroy the tumor. And actually all what it takes, learning from the past, one approach is not going to cut it, because actually the tumor can make mutation and phenotypes to build the resistance against your single approach. You have to attack it by simultaneously, immediately, by multiple polyvalence, a multivariant. And nanotechnology can enable us to have that multifunctionalization, and we can eradicate the tumor. And this is our goal in the center, eradication. I know I have five minutes left. And we can take advantage of the difference between normal blood vessel versus tumor blood vessels and certain receptor being expressed on the tumor blood vessels. So we can actually take alpha V beta 3 integrin, which is highly expressed on endothelial cell in new blood vessel around the tumor and the cancer cell itself. And we were able to functionalize the polymer BLGA and conjugate in alpha V beta 3 ligand into the surface. And we can encapsulate a chemotherapy inside. We discovered even this by itself has a major impact on the cancer. So we have a lot of patent on this. We have about 35 US patent on this platform. And that's led to a company called Nanopharmaceuticals. They are going to glioma at MD Anderson and the pancreatic cancer at Cleveland Clinic, clinical trials. So this is the characterization of the nano. We have publication in terms of multiple tumor types can be easily attacked and targeted by this. And we showed, I'm going to show you here very quickly, in terms of targeting chemotherapy, many chemotherapy, cisplatin, doxorubicin, TMZ, temolazamides, into the site of action. So here is a cisplatinum, in nano encapsulated inside this nanodiaminotetract targeted treatment. You can get about 100 nanogram per gram tissues relative to cisplatinum. You get about 5-fold increase in the chemo uptake. And this is actually, I'm doing it at the end of the study. If I do it and at earlier in the study, you get higher accumulation. But this is a net accumulation, 5-fold greater accumulations with the nano-targeting. And actually, we have seen the side effect on the cisplatinum because you can get peripheral neuropathy with the nano-targeting because now you are shuffling everything into the tumor, less into the nerve. You can have a normal animal walking around. I have a lot of these images. This is under review. This work is under review. You can deliver paclitaxel into pancreatic cancers. You can see the differences here. It's almost about a 20 nanogram. You get about 160, about 8-fold increase. You can do this for doxorubicin. You can do it for many chemotherapy. So the question, nanomedicine can really help us to improve the PK and PD efficacy and safety. We can, for diagnostics, we can improve sensitivity and specificity for early detection. Imaging, you can use an RGD here, and you can bond it to the the polymer. You have multiple of this. You can conjugate it chemically to a DOTA complex, book gadolinium for MRI imaging. I'm going to skip these. The whole idea, if we look at the status of medication and responsiveness, many classes of compound here in many diseases, you can see the percentage of non-responder. There is about one-third to two-thirds of the patient. Only one-third of the patient respond very well. And the one-half to two-thirds do not respond. So this can be easily, nanotechnology and by targeting, can enable us to get into the site of action and improve the efficacy, lower the dose, and improve the safety profile. I don't have much time to talk about precision medicine, but this is just an example to show the state where we are and how can we improve this. There is a lot of future application of medical technology. We experiment DNA sequence for routine checkup. We predict actually because the price tag for sequencing is coming down to $500 when it becomes $200. Actually, the primary physician is going to be doing the DNA sequence for you and having the genomes and will be stored in a chip. I'm almost done. This is almost right. You can match tumor to, and this can be stored, and this is going to happen very soon when the price comes to $200. Matching tumor to drugs, letting your body, the immune therapy is a big hype here and actually, there is a lot of good outcome. Gene therapy by sensors and auto medicine delivery simultaneously. Improve healthcare delivery, robotic surgery, biocompatible part. This is my final slide. It takes imagination. You look at this fellow here, the artist is looking at what? The egg. What is he drawing? The bird. For the young people, you have to have that imagination. You have to be a visual guy. Imagine. But more, as important as imagination, you have to execute. There are a lot of dreamers, but they never act on their dream. You have to execute. The extent to which nanotechnology will have an impact on our lives only depend on the limits of human imagination and execution. They have to go hand in hand. With that, I'd like to thank many people within my groups. I am actually a bridge builder. I collaborate with people around the U.S., many institutes around the globe. Even in every country, I will put a dot. I have a map. I put dot on every country around the world. I have collaboration. I have graduate students from a lot of places, and I love to mentor and train people and help people. So along with that, at the same time, collectively with these people, we were able to move things from the bench to the bedside and hopefully to the human use. Thanks so much for your attention.